Hey guys, and welcome to our Exam 5 study guide. So this review video is only going to be comprised of chapters 13 and 14, but do remember that your Exam 5 is your comprehensive final exam. So all the other review videos prior to this can be found on Canvas, and that is what will aid you in studying for the final. All the review videos, all the other previous study guides, PowerPoints, everything. So this is only going to cover the material since exam 4, which is only chapter 13 and 14. But don't forget to look at the supplemental material that is posted, and then also any review videos, study guides, also previous exams that can also guide you in topics that are going to be important for studying. Okay, so just like our last videos, I'm going to use the study guide as kind of like a backbone to this video. So let's go ahead and just jump right in. So the first one, I'm actually really not going to go over too much, but I am going to tell you where to find this material. And this material can be found on our Canvas page. So if you were to go to your modules, you would go to our week 13 module once it loads, and it will be under the history of DNA. So under this page, you will find some videos or also some interactive stuff that will aid you in the history of DNA. We did not cover this in class, but it is still fair game for the test. So with that, we have some interactive videos, animations right here that goes over all the big scientists that help discover DNA. Also for DNA replication, that will also aid you in the process. And then also some reading material, which is exactly the same thing that is in the video, but if you prefer to read over watch, then you can definitely do that. And then also I do have a short PowerPoint that goes over it. So I have it in 3 different ways, guys, where you can learn all this information, so whichever one helps you. But this is where you can find all of it, but based on the study guide, it says that you need to know the person. So you need to know what experiment goes with what person, what was their experimental design, so what did they actually do, and then what was like the outcome, their hypothesis, and then what was the final like aha moment from the experiment. So kind of like what was the result. So definitely that will be testable material. So that's just for number 1. So for number 2, it asks for what is the structure of DNA, draw the AT-CG base pairs, Watson and Crick, and then different evidence that were there, and then what does it mean when DNA is anti-parallel. So to answer that question, it can be found in our chapter 13 lecture. So let's go ahead and pull that up. So chapter 13, so the DNA structure. So like we talked about very long time ago at the beginning of term, was that DNA is a helical structure. It's double helix. So if you were to take a ladder and twist it, it would kind of look like that. The backbone of it, so the outside part are sugars, and then the inside part are all like the nucleotides and everything. The backbone itself is held together by phosphodiester bonds, whereas internally, where all the base pairs are, the nucleic acids, so the adenines, thymines, guanines, and cytosines, those are all in the center, and those are held together by hydrogen bonds. So remember that A always pairs with T's and C's always pair with G's, so meaning that a pyrimidine is always with a purine. It's never a purine and a purine or a pyrimidine and a pyrimidine. And let me show you why that's the case. So if you have a purine and a purine together, they are, what's it called? A 2-ring structure, so that means that it's too wide. So it's not very even, it's not very stable. If you have a pyrimidine and a pyrimidine together, it's too narrow, so there's too much space in between them. But if you have a purine and a pyrimidine, then you have just the proper amount of space between them, and they also counterbalance each other, and they're able to form that hydrogen bond in between them. So this is what they look like. Whoopsies. So adenine and thymine, so a double bond to I'm not double bond, I'm sorry, a double ring to a single ring, and then guanine is a double ring to a single ring of cytosine. So that is how they go together, and then whenever they actually come together, they form that nice little hydrogen bond between them. And that's why it's so important, because hydrogen bonds can only go so far and can only be so close. And then this is our double helical structure of DNA. Okay, let's see, what else is there? How did Watson and Crick figure out this structure? So that can be found underneath the history of DNA, but I will mention it very, very briefly. So how they figured it out was actually through Rosalind Franklin's experiment. So Rosalind Franklin, she did X-ray crystallography. I do want y'all to know that. But what it was that she found out that DNA was a double helix structure based on the X-ray crystallography. And if you read over it and watch that short little video on it, you can definitely see the image of the X-ray crystallography that sort of shows an X. And actually, I'll go ahead and pull it up real quick. So I'm just gonna Google a picture. So what Rosalind Franklin found out was this structure. So this was the X-ray crystallography image that she got after the experiment, and that is that X structure, that double helical structure. So if it was to go bigger, it would actually bubble up, kind of like what we saw in this. So this is what we are seeing. We are seeing this part in the X-ray crystallography, that X part. And Watson and Crick, they took that idea and then they came up with the idea that it was that layout. So what they did was that they realized that that the DNA was made up of two chains of nucleotide pairs that encoded for the genetic information. So Watson and Crick did not physically do the experiment. It was Rosalind Franklin who did. And they won a Nobel Prize over it. And it kind of sucks because Rosalind Franklin was not able to get it because she died before it was awarded. And Nobel Prize winners can't be dead when they get it. So it kind of sucks. But anyway, so with that, what does it mean when DNA is anti-parallel? So that can also be found in our lecture. So what does it mean for DNA to be anti-parallel? So it means that the strands will run in opposite directions. So meaning that this is one side of the strand, the 5' to 3' end, and then the other strand of DNA is a 3' to 5' end. So they are backwards of each other. So each strand has a 5' and 3' end, and then one end, specifically the 5' end, always has the phosphate head, as we see right here, whereas the 3' end has the sugar end. And that's what it means to be anti-parallel. So they are going to be opposite of each other. And this is very important whenever it comes to adding nucleotides. Okay, so with that, let's talk about number 3. So number 3 is, how do we know DNA replication is semi-conservative? Make a table of all the enzymes involved in DNA replication as well as their role, and build a concept map of the steps. So meaning just know the steps, what happens during each part of DNA replication essentially. And the words that are going to be very important to number 3 can be found at the bottom. So right here, the word bank for DNA replication. If you know all these words and all these concepts and can apply them to questions, then you should be good to go in accordance to DNA replication. But let's go ahead and go over it very quickly. So DNA replication, there are some animations in our lecture that you can definitely look at. But let's talk about the first one, where DNA is semi-conservative. So DNA itself, or replication itself, is semi-conservative because you always have half new and half old. So we have our DNA right here, and then whenever we replicate it, we create that bubble that we talked about. And then what we do is that we use the original DNA as a template in order to create our new one, which is the red. And then once we double or replicate our DNA, we get part old and part new. So the old is blue, red is new in this case. So that's what it means to be semi-conservative, meaning that it's not entirely new when we replicate our DNA. Okay, so with that, let's go ahead and talk about all the enzymes, all the big players that we need to know. So here's the vocab. So there are 7 main ones that we talked about in class. We talked about helicase, topoisomerase, single-strand binding proteins or SSBs, primase, DNA pol 1 and 3, sliding clamps, and DNA ligase. So definitely need to know when all of these enzymes or proteins come in during DNA replication, as well as what is their function. So for the most part, I have them relatively in order as to when they appear, but some of them kind of appear at the same time, at the same stage. So let's go ahead and talk about DNA replication. Oopsies, wrong way. So let's look at this. Actually, let me just pull up an entire picture so it's easier. So let's look at this guy. So this is the entire DNA replication process. So the first thing that happens is that we get the signal to start replicating. And then once we get that signal, what's going to happen is that we are going to create this bubble. And then once that bubble comes in, actually I don't like this picture. Just kidding. I'm going to go to the one in the lecture. So what's going to happen is that topoisomerase, primase, and helicase, and then as well as SSBs, they are going to be present in the initiation of DNA replication. So what's going to happen is that topoisomerase, he will relieve the strand. He relieves the strand caused by all the tightly wounding. So he aids in essentially giving support to the strand to help all the stress that it's about to be put under. And then we also have primase. So primase specifically will add that RNA primer to the DNA. So that is kind of the initial point of DNA replication, where later on DNA polymerase will come in and start replicating. And then we have helicase. Helicase is a specific enzyme that will untwist our DNA. And then at the same time, we have these single binding proteins, which will bind and stabilize on both sides of the DNA, which was unstranded, to provide support, kind of like topoisomerase in a way. So these single binding proteins, or these SSBs, what they do is that they kind of anchor the DNA, and they also keep it from coming back together slightly. Okay, so that is the initial part of DNA replication. So that is the first thing that will happen. And then what will happen is that we're going to start building something called the leading strand. So when we're building the leading strand, this is where we are making actual new DNA. So this is where we are actively replicating it. And then what's going to happen is that, let me actually just go to the picture. So what's going to happen is that we are going to have DNA polymerase come in, but DNA's polymerase will not come in if primase or that RNA primer has not been added. So DNA polymerase, he cannot synthesize the leading strand or the lagging strand, for that matter, if that RNA primer has not been added. But let's say it has been added. So let's say we have our RNA primer. So that RNA primer, it will allow DNA pol 3 to attach to the DNA strand, and then from there it will be synthesizing in the 5' to 3' direction. So DNA pol 3 will specifically add to that 3' end of that DNA polymerase. I'm sorry, not DNA polymerase, of the RNA primer. So DNA polymerase 3 will bind to the 3' end of the RNA primer. And then from there, he will continue to replicate along this strand in a continuous motion of the 5' to 3' direction. So that's for the leading strand. Now let's talk about when we're building the lagging strand. So if we're building the lagging strand, which is going to be on the other side of the bubble, we are going to be going kind of in the wrong direction. So the other strand is not built in a continuous motion. So let me see if I have a better picture. So here's a bigger picture. So this is the leading strand because it's in one single motion, whereas on this side we can see that there's some gaps, and this is called the lagging strand, and those gaps are called Owazaki fragments. So what will happen is that DNA polymerase, he will also attach to where that RNA primer was. But what will happen is that he will move along the strand, but like in hiccups, like he'll just hop, and then he will slightly let go, which is where those gaps occur, and then reattach, and then continue synthesizing. But there will be some gaps, and like I said, those gaps are called Ozaki fragments. The leading strand and the lagging strand, they are built at the same time, just the lagging strand is built a slightly slower. But in order for DNA replication to be complete in this part of the DNA, we need something to come in and tie those Owazaki fragments together. And that is where DNA ligase will come in. And DNA ligase, he will come in and rejoin those fragments. So it's a continuous strand like the leading strand. I believe that's pretty much the big picture for DNA replication. So in our lecture, we do have other figures that you may like better than the ones that I focused on in class or in this video. But also, like I said, there are a lot of videos that can go over this if you want to solely focus on that. Okay, so let's go ahead and go to our next question. So our next question is, how does the cell correct errors if errors occurred during the copying process? So it happens where sometimes replication, there are some mistakes. We try to be as perfect as we can, but that doesn't always happen. But we do have some proofreading mechanisms. So DNA polymerase, he will also proofread and he'll fix his mistakes if he can. But sometimes the mistakes, they're not caught right away, or the DNA is actually damaged later on, such as like through X-ray or UV radiation, such as like the sun. So whenever that happens, there's something called nuclease. So nuclease, he specifically will come in and cut out the part of DNA that is damaged or that is paired incorrectly, as we see right now in the figure. And what he'll do is that he'll cut it out. DNA polymerase will come in and replace that section or that base pair, and then DNA ligase will come in and tie together that section so it's continuous once again. So but for the most part, we're pretty good at doing this. But like I said, there are some cases where we'll have UV radiation create a dithymine dimer, which can lead to skin cancer if left unchecked. And then what will happen is that nuclease will also come in, take out that damaged section. DNA polymerase will put in the new section, and then DNA ligase will come in and retie it together so those thymines are bind to those adenines rather than bound to each other. But those are pretty much the basics what will happen. So the big three enzymes that are a player in correcting replication error is nuclease, DNA polymerase, and DNA ligase. Those are the big guys. Okay. So with that, let's go ahead and talk about number 5. So number 5 is about the Beetle and Tatum experiment, or that one gene, one enzyme hypothesis. So that is found in chapter 14. So chapter 14, let's go ahead and go to that thing. So chapter 14 is about gene expression. So with the Beetle and Tatum experiment, I do want you to know what organism did they use, and it was Encrasia, or a common bread mold. And the reason why they used it was because it's pretty simple, and also because it can create progeny very, very quickly. Like its life cycle is very, very quick. So they don't have to wait months on end for it to grow. It pretty much grows for the most part overnight. Um, but what they came up with, or their idea was, if genes were connected to enzymes, um, then that means that they could potentially induce mutations, which is what they did in their experiment. So, what they did was that they took spores from the bread mold and they mutated it using X ray or radiation. So, with that, they created some mutants. And then afterwards, they took the descendants or like the progeny, so the mutated bread mold, and they grew them individually into different, not different growth mediums, in minimal growth mediums in order to grow them. And then what they saw was that for the most part, each spore or each bread mold was able to establish a growth colony. So they were able to continue to grow, but there was some that were not able to, as you see in this image right here, where you have growth, growth, but then no growth. Okay, so that was the first leg of their experiment. So what they wanted to do was to determine what happened in this part where there was no growth. And that leads us to this. So what it was that in order to figure out what was broken in that mutant, like what happened here, like why was this not happening. So what they did first was that they grew each mutant, so they grew each one, and they each gave them a full set of like vitamins. So they supplemented their medium. So they either gave them a full set of vitamins, or they gave them a full set of amino acids. And from that, what they did was that they saw that it was amino acid based. And with amino acid based, we know that amino acids code for proteins and enzymes are protein based. So they continued with the amino acids. And then from there, what they did was that they individually added each amino acid to each test tube. So as you see right here, they had all 20 right here, but they wanted to know which specific amino acid was lacking. So what will happen is that they took the mutant and then they added the mutant to each individual one that only had one amino acid in it. And then based on which one grew, determined where the mutation occurred. So in this case, AGR was what was missing. So the mutant grew. So that is how they came up with one gene, one enzyme idea. So it was a very lengthy process, but actually a very simple experiment. And I believe they did win a Nobel Prize for it, actually. So kudos to them. But I do have that Bill Nye video that probably sums it up a little bit better. And, like, come on, who doesn't like Bill Nye? So definitely take a look at that. Also, you can do some Googles, but for the most part, everything that I mentioned is what I want you to know in accordance to the Beadle and Tatum experiment. Okie dokie. So with that, it says make a chart or a concept map about the steps of initiation, elongation, and termination in the context of transcription. So meaning, kind of like with the DNA replication, if you know these words right here and what do they pertain to in the context of transcription, you are good to go. But let's go ahead and talk about it real quick. So that can be also found in chapter 14. Oops, wrong way, right here. So let's go ahead and talk about, we're going to talk about it in terms of eukaryotes. So in terms of eukaryotes, there are three main steps for transcription and translation as well. So the first step is initiation. So let's talk about initiation. So initiation, this is where the whole process will begin. And what will happen is that there's something called a promoter region, but in terms of eukaryotes, there's something called the Tata box. I do want you to know the Tata box. So this, the Tata box is crucial in forming the initiation. So if a Tata box is not present, then transcription will not occur. But what will happen is that RNA polymerase 2 will bind to that promoter region. Also, there are some transcription factors that will aid in binding. So as we see right here, we have some transcription factor present on top of the promoter that will aid for RNA polymerase to actually bind. It's more or less just kind of like a guiding system for RNA polymerase. But then once RNA polymerase has bound and has bound to the DNA, what will happen is that it will unwind the strand. As we see right here, we see that bubble that we always see. And then from there, RNA synthesis can begin. So meaning we can start transcribing some RNA from our DNA. So that's the initiation process. Let's talk about elongation. So elongation is where we are actually building our RNA from our DNA. So RNA polymerase will travel down the DNA, and as it moves down, it's adding RNA nucleotides in the 5 to 3' direction and building that RNA. So normally this is roughly 10 to 20 base pairs at a time, kind of just depends. For the most part, it's around 10 to 20. And then as it's traveling down the line, it's going to keep going and making RNA until it gets a termination. So this elongation is very simple, very routine, and the same. What's going to happen though, in terms of termination, eventually it's going to get a signal to stop, and that signal is going to be something called a stop codon. It's going to be a specific sequence, and we'll talk about that later on, but it's a specific sequence. It's either actually no, just kidding, we'll talk about that in translation. Anyway, just know that it gets a stop signal, and it will get the signal. We'll go with that for right now. It'll get the AAU signal, and then that will tell RNA polymerase like, oh, I'm done, and it will let go or fall off. And then that's the end of transcription, but there is one more step that can happen before translation can occur, and that is the modification of what we just made. So in order to modify, what we need to do is that we need to add a 5' cap and a poly A tail. So that 5' cap is phosphate heads, and then that poly A tail is a bunch of adenines. It can range from 50 to 250. It kind of just depends. I honestly don't know what determines the number, but I do know that they have to be added to the 3' end. And then once that is added, what's going to happen now is that we are going to do some exon and intron modifications. So with that, we have coding and non-coding regions. So non-coding regions are introns, and these are the part of the RNA that we don't care about. So what's going to happen is that we're going to have some spliceosomes come in and literally cut out those introns, and then they're just going to be discarded. And then the coding regions, which are the exons, are going to be spliced back together, meaning that they are going to be sewn back together to make all one long coding segment. And this is the RNA that can be exported out of the nucleus into the cytoplasm of the cell. So this is what we want. And that will be the end of transcription. So with that, that's going to bring us to translation, if I remember correctly on our review guide. So yes, it's going to bring us to translation. And then right here, all of these vocab is what I want you to know in the context of translation. So let's go ahead and talk about translation. So that's also in chapter 14. So let's talk about the first process. So the first process is initiation of translation. So this is where mRNA, so this is what we exported out of the nucleus, is mRNA. This is where a small the small subunit of the ribosome will attach to the mRNA, and then once that small unit of the mRNA is attached, the larger subunit of the ribosome will come in and also attach, creating a complex. And then that will happen whenever it comes across something called a start codon. And that start codon is AUG. And a bad way of remembering it is that school starts in August, so AUG. So it's definitely very, very important, that start codon, because it will establish the reading frame, meaning it will determine how long will that ribosome translate the mRNA. So that is the start process. Okay, now with that, we're going to go to elongation. Elongation is in 3 parts. So the first part is something known as codon recognition. So codon recognition is where tRNA will come in. So this is the one that will actually start adding on the amino acids. He will come in to the A site of that ribosome complex, and he will add the matching anticodon to the mRNA. And then once that happens, we get something called the peptide bond formation. So once that codon recognition has occurred, the A and the P site of the ribosome are occupied, and at that point is where that peptide bond will occur between the amino acids. And then that is how we actually start getting our peptide chain formed. And then we get something called translocation. So after that peptide bond has formed, the P and the A site are occupied. But we need that A site open in order to come back and add more amino acids to our chain. So what's going to happen is that the ribosome is going to shift, and then with it shifting, what is in the P site is going to go into the E site and exit, and then what was in the A site is going to go into the P site. So now all we have is the P site occupied, the A site is open so it can accept another codon from tRNA, and then the E site is also open because it discarded the tRNA that was not necessary anymore. And then this process will keep going on and on and on until it gets a signal, which is a stop codon. And there are 3 stop codons that I want you to know. It's UAA, UAG, UGA. So you definitely need to know those 3 codons in terms of termination. But what's going to happen is that it will come across one of those codons. It doesn't matter which one, but it will come across one of them. And then it will cause a chemical reaction which will make the ribosome subunits fall off. So it will make the ribosome fall off of that mRNA, and then we're going to have a protein chain floating around. So termination, very simple. So what's going to happen is that protein chain is going to be released, as well as the mRNA is going to be released, and then that ribosome subunits will detach from the mRNA. Okay, that is all for translation. So now let's talk about mutations really quick. So let's go to mutations. So mutations themselves, they can be divided up into 2 groups, which are known as substitution or insertion/deletions. So with substitutions, there are 3 main divisions, I guess, and they're called silent, nonsense, and missense. So with silent mutation, what happens is that there is a slight change in actually, I don't think there's any change. Yeah, just kidding. There's no change. Just kidding, guys. So there's no change in making the amino acids. So as you see, we have our DNA, we have our mRNA, and at the same time, it still makes the same amino acid. So what happened in terms of silent mutation was that in this case, this C, which is this is our non-mutated DNA, That C got turned into a T, but even though it changed our mRNA sequence, it still coded for the same amino acid, meaning that it did not hurt anything. So it's kind of a pointless mutation. Didn't cause any harm. Now, whereas in a missense mutation, it can be detrimental depending on what gets mutated. With missense mutations, what happens is the same thing. In terms of silence, where we start off with a C, but then it became a G. But in this case, it changed our amino acid. So we originally were coding for this guy, but then it got coded for this guy. So that's a missense, meaning that we get an entirely new amino acid based off of that mutation. And then a nonsense mutation is where it changes the amino acid to a stop codon. So meaning that we will terminate our translation process early, which is not good, because sometimes that can lead to incomplete or a non-functional protein, which is not what we want. We want all of our proteins to be functional. Now with insertion and deletions, let's talk about that. So insertion is exactly what it sounds like. We inserted something. So something was inserted in the original sequence, and in this case, another C was added. So with our insertion, it will cause something called a frame shift mutation, meaning that reading frame that we started with, where we had our start and our stop, now that reading frame is slightly shifted. So with our reading frame shifted, we can either make our reading frame shorter or longer, which is not ideal. We want it perfect so we can have good proteins. Now with deletion, it's exactly what it sounds like as well. We deleted something, so we took something away from our original sequence, and that will also shift our reading frame as well. So either way, with insertion and deletion, you're going to have that reading frame will be altered no matter what in terms of insertion and deletion. And I believe that was the last concept on our review. It was fabulous. Okay guys, so like I said at the very beginning, this just covers material since our exam 4. All of the other material can be found on Canvas along with supplemental material. Don't forget to look at your old test, look at everything that's posted on Canvas. I can't stress that enough, guys. Also, this was not an exclusive list of everything. It was kind of just to point out all those highlights. If you have any questions, please feel free to contact me. If not, I will see you in class, and have a good night.